Researchers from the University of Illinois at Chicago have discovered zombie brain cells that come alive and grow after death. In a study that has recently been published in the journal Nature, researchers conducted gene expression analysis in fresh brain tissue collected during routine brain surgery. This tissue was analyzed at multiple points after it had been removed from 0 to 24 hours as a means of simulating the post-mortem interval and death. Gene expression in certain cells was actually found to have increased after death, growing to gargantuan proportions. This gene expression was found within specific inflammatory cells called glial cells. These cells differ from the nerve cells and don't directly participate in synaptic interactions and electrical signaling. Instead, as per the book Neuroscience 2nd edition, glial cells provide supportive housekeeping functions that help define synaptic contacts and maintain the signaling abilities of neurons. While carrying out the study, UIC researchers discovered that glial cells grew and sprouted long arm-like appendages for many hours after death, challenging previously held assumptions about what happens to the brain after death. The pattern of post-mortem changes in the glial cells was found to peak after approximately 12 hours, increasing at the same time that neural genes were ramping down. In a press release, Dr. Jeffrey Loeb, head of neurology and rehabilitation at UIC's College of Medicine, said it wasn't too surprising to note the post-death enlargement of glial cells as they are inflammatory and work to clean things up after brain injuries like oxygen deprivation or stroke. However, Loeb was a corresponding author on the paper, noted that the implications of the discovery are indeed significant. This is because the majority of the research studies that use post-mortem human brain tissues to look for treatments and possible cures for conditions such as schizophrenia and Alzheimer's disease currently don't account for the possibility of post-mortem gene expression or cell activity. Loeb explained most studies assumes that everything in the brain stops when the heart stops beating, but this is not so. Our findings will be needed to interpret research on human brain tissue. We just haven't quantified these changes until now.